Hi, it's Thursday morning. We're Lab Group 6B. This is Lab 12, Color, Vision, and Acuity. And in today's lab, we're going to be examining the human optical system of the eyes and the brain.
So I'm going to explain eye dominance, our first test. So we have our implement, a piece of paper with a hole in it. Step 1. Place piece of paper in front of face. With both eyes open, find a target in the center of the hole. After this step is completed, close each eye. Your dominant eye is the eye in which the object stays in the hole when you close your eye. Your non-dominant eye is the eye in which your field of view disappears when you close your eye. Next we're going to talk about finding the blind spot in the human eye. So we have our test. Test is a piece of paper with 2 circles on it. So what we're going to do is we're going to place the target almost on our nose with one eye closed. As we move the target out, we will begin to see the one circle disappear from the field of view at where the blind spot is located. For Alex and I, this is located approximately 15 degrees off-center in each eyeball.
Alright, next we measured the field of view of the eye. So here we have a whiteboard. It's 96 inches across. And what I did was I closed one eye and tried to see how close I could stand to the whiteboard and still be able to barely see the edges of it. That turned out to be 18 inches away. Then I did the same thing with both eyes open and was able to stand 5 inches away. This corresponds to 157 degree field of view for one eye and 174 degrees for both eyes.
And now for an eye test.
Alright, so I'm 20 feet away from the eye chart on the wall over there. And the way this works is I try to read as many of those lines of letters as I can. And whichever the smallest one is that I can read correctly, that corresponds to my vision. And since I'm 20 feet away, if I can read the one that corresponds to being 20 feet away, then I have 20/20 vision. If I can only read a bigger one, I have worse vision. Or if it's a smaller one, I have better vision. And we just tested, and I was able to do 20/15 vision. So we'll go ahead and point the camera at the board, and we'll see if I can do that again. The 20/15 line is line number 9. LESODPCT.
Excellent.
Is that right? Alright, one more thing about the eye test is that the dimensions of all these letters is very important. If we look at the E, for example, we notice that it's 5 units tall, and that each dimension is a unit. So like this length here is 1/5 of the whole height, or this length here is 1/5 of the entire width. And that's the same for all of the letters down the chart.
So now we're going to run a contrast sensitivity on each of our eyes. We're going to do this by using an online application which will flash bars at us and vary the contrast as we proceed through the test. We'll start. If we can see the bars in the target in one frame versus the other, we select the frame that we think we can. In this case, it was clearly the second interval. We're going to present stimuli again. This time it was clearly in the first interval. And as you can see, the program gradually reduces contrast each and every time we do this. This is useful in determining the contrast sensitivity of our eye. Alex and I both did this test, and we were able to get down to less than 0.3 on the contrast scale with a spatial frequency of 8 cycles per millimeter.
So in this part, we're testing our flicker fusion frequency, which is the fastest frequency that this little light blinks, and we can still detect it with our eye. So what we did was we had 3 LEDs, a red one, a green one, and a yellow one, and we would put it at 5 different voltages, and then we would increase the rate at which it flickers until we can no longer detect it with our eye. And what we saw was that with the green and yellow LEDs, we were able to detect faster flicker than with the red for both Colton and myself. And as we increased the voltage for all 3, we could also detect faster flicker. And also for all 3 colors, Colton could detect faster flicker than I could, which is totally lame.
The next portion of the experiment was to actually test the visual wavelength over which our eyes are sensitive. In order to do this, we have a grading monochrometer here. And what we do is we look inside of the monochrometer, and we have an incandescent bulb which has all of the wavelengths. It's this white light, and we move this. We rotate the knob until we see different colors. And now you can see it went to red. It's going a little red, and it's starting to actually fade out completely. So as you can see here, this would be the actual limit or wavelength detection of the camera, is when that light completely disappears. Alex and I both thought we'd be around 400 to 700, but we were pleasantly surprised to see that we were around 380 nanometers to about 740 nanometers.
So lastly, we're going to explain stereoscopic vision, and this allows us to see 3D effects even with 2D images without the fancy schmancy glasses. So the trick is that you want to take one of these images, and normally when we just look right at it, both our eyes are just focusing on one spot, but what you want to do is focus through it so that our eyes are looking through it to different spots. You see this image, it has kind of a periodic nature to it, and so what you want to do is get your left eye to focus on one of the periods, or look through so that it's looking through one of the periods, while your right eye is looking through the other period. And when your brain overlaps these two images, when it processes it, that's what causes the 3D effect. It's the difference between the two things your eyes are seeing. That's it for Lab 12 and the semester. This is Alex and Colton. Goodbye.